Share screen. There's sound. I will help myself. Hi everyone. I hope you can hear me now. I hope so. So as he told, I come from the Center for Music in the Brain in Denmark, and I'm very happy to be here today to participate in this journal club. And I will talk about tapping the beat of 4 subdivisions, neural entrainment, musical training, and the binary advantage. So let me start a brief outline first. I will start just introducing what is the beat and what are subdivisions. And then I will quickly jump in the study about behavioral and neural responses to subdivisions. So imagine COVID-19 is over and we can go back to nightclubs and have some party. After a few seconds listening to music, we start detecting regularities. These regularities allow us to entrain too. So we can move the head following these regularities, tap the foot. This is called beat. And we detect these regularities because of beat perception. But this is not the full story, because we can organize these regularities, this beat, in groups of 2 or 3, or we can subdivide the beat in faster levels, which would be like the subdivision level. So we have beat perception, and then we have the metrical structure in which we organize the beat in faster and slower levels. For our talk, we will focus on the subdivision level. So this is faster rates compared to the beat. So we are not the first studying beat perception and subdivisions, of course. And previous research found that when we add subdivisions, when someone taps to the beat, this facilitates the motor synchronization to the beat. So when we add tones between taps, the tapping asynchronous decrease. This is known as the subdivision benefit. And it's speculated that this happens because it reduces the underestimation of inter-onset intervals, and in turn improves the prediction of future events. So Zendel and colleagues, for instance, found that when we add additional tones between taps, like here, like 1 to 2, when you add an additional tone duplets, if you add additional tones triplets, you add 3 additional tones, you have quadruplets. So when you add these additional tones between the tappings, you get a reduce of the mean asynchronous, as you can see here in the graph. The black bar is for the beat only alone when there are no intermediate sounds. And then you can see that the mean asynchronous decrease. And this can happen at different tempo. This is part of the subdivision benefit. Also found that there is a decrease in variability. So this is the standard deviation. And also for duplets, triplets, and quadruplets, you have less variance for tapping to the beat alone. This is not the full story. There is also some studies that try to understand how subdivision affects also in relation to musical training. So Bruno Rep in 2007 checked the different sensorimotor synchronization thresholds using different subdivisions, 9 of them, and compared musicians and non-musicians. Here, the black line is for musicians and the dashed line is for non-musicians. And these are the different subdivisions. Divided into 2, 3, 4, 5, 6, 7, 8, and 9. And what he found is that there are higher synchronization thresholds, so people could not tap directly to faster tempo for quintuplets or septuplets compared to other subdivisions. And also he found that there was an increase of variability dependent on musical training. So musicians had more variance and lower thresholds for the synchronization to triplets. So all these studies inspired us and made us thought that maybe the subdivision of the beat and its sensorimotor benefit could be related to some properties of the oscillating neural system. Maybe these oscillations enhance particular harmonics of the frequencies entrained to the beat, like the binary one, the first harmonic. So to test this, and we expect to find differences in the binary and non-binary subdivisions in the amplitudes of the frequency spectra of the EEG, and also in the accuracy and consistency of the tapping. As well as Bruno Rep showed, there are differences because of musical training, so we hope that these responses will be sensitive to musical training. In our methods, to collect the neural responses, we used a 32 electrode active cap. Also recorded the tapping, the behavioral responses using a MIDI device. And finally, we selected a group of two groups of participants, 15 non-musicians and 16 musicians. We created a stimuli, the beat, which was 30 milliseconds long. and it was presented at every 800 milliseconds. This is at the frequency of 1.25 hertz. Also, we created 4 subdivisions: duplets, triplets, quadruplets, and quintuplets. So this is the tone used to create the bit, the 30 milliseconds one, and then we use the same tone, we add it in the middle of the cycle to create the duplet. If we add 2 additional tones, we get triplets and quadruplets and quintuplets. In our procedure, we started first putting the participants in a rhythmic discrimination task, which is the musical ear test, but only for the rhythms. So they had to discriminate between pairs of rhythms that were the same or different. And we found that musicians had higher scores compared to non-musicians, so good. Then the participants went to a practice trial in which they learned the paradigm that I'm going to present in the next slide, and finally the test. So this is our paradigm. Our paradigm consisted of 36 auditory sequences, 9 for each one of the 4 subdivisions, that could be divided in 4 sections of 20 seconds, containing 25 bits each. So this is the paradigm. Participants first had to listen to a beat, an isochronous periodic beat. After the 25 bits, we start with one of the 4 subdivisions, duplet, triplet, quadruplet, or quintuplet, then the same beat again, and finally the same subdivision again. But in the last section, participants had to tap to the beat of the subdivision. So let's listen to one example, for instance, the triplets. This is a shortened version with only 4 bits. Let me play. And now, so they had to do that 9 times for triplets and 9 times for every subdivision. What was interesting of this paradigm is that it allowed us to compare what happens in bit perception before and after listening to the subdivision. So if there is any effect of the subdivisions on bit perception, comparing the section 1 and section 3, and also what is the contribution of the motor tapping task to the bit, because we can compare listening to the subdivisions alone versus listening to the subdivision while tapping to the bit of that subdivision. Now let's move to our responses, to our results. We computed the average mean for every subdivision and trial of the tapping responses, and we placed them on a circle in which it was reference of 800 milliseconds, so the full cycle of the beat. And then we separated musicians and non-musicians in these circles, and the 4 subdivisions: duplets, triplets, quadruplets, and quintuplets. We can see here is that there is every little circle on the circle represents a participant accuracy, and the 0 phase, the angular position, the 0 is when the sound was presented, the beat. So where the place where we were expecting the tap to occur. As you can see here, and thanks to William Watson test, we found that non-musicians quintuplets, the tapping to the quintuplets was different from the ones in musicians, were more dispersed. And also we found that also these quintuplets in non-musicians were different from triplets and quadruplets and almost duplets. This was regarding tapping accuracy. But what is interesting here is that we can use the vector length for every participant. As you can see in the circle, there is line, the radium, and this is an inverse of the variance of the tapping. So the longer the vector length, the more consistent the tapping was. And these are just repeated measures Anova between musicians and non-musicians, and we can see that musicians had more consistent taps than non-musicians in general for all subdivisions. However, we can also see that in both groups, musicians and non-musicians, the taps related to quintuplets, the darker gray bars, were less consistent compared to other subdivisions. Finally, we found a very interesting result, that is that for non-musicians, the taps to the beat of the duplets were more consistent than the others. So now let's turn to our neural responses. We recorded the EEG through electrodes. We averaged the trials that corresponded to the same subdivision rhythms. And then we decomposed all these temporal EEG electroencephalograms into their frequency dimension. So we could see how different steady-state evoked potentials appeared and corresponded to a peak in the frequency spectra. These peaks allowed us to look for the amplitudes at the frequency of the beat and also the frequency of its metrical grouping or its subdivision. The thing was like this. Here there is an example, for instance, for duplets. You can see that the auditory sequence on participants were listening to the beat, then as two they were listening to the subdivision, listening to the beat again, and listening to the subdivision while tapping to the beat. And here is for all the subdivisions: duplets, triplets, quadruplets, and quintuplets. Blue lines are for non-musicians, and red lines are for musicians. You can see during the first three tasks, there are not much differences between musicians and non-musicians, but visually you can see that the peaks increase much more for musicians than non-musicians during the tapping task. Also notice in the top of plots that there is the frequency, the neural responses to the tapping frequency greater in the contralateral premotor areas, and also maybe in the ipsilateral cerebellar areas. That's also very interesting. Okay, now let's do some statistics on that. We compared first what happens to beat perception before and after listening to the subdivisions. We ran 5 three-way mixed designs Anovas for each frequency of interest. And we found only a main effect of task, no main effects of subdivision, neither rhythm or group. And this summarized the results, the post-hoc comparisons. We found that after listening to all subdivisions, there is an increase at the beat frequency in neural entrainment at the beat frequency, and also an increase at the frequency of its first harmonic, which is related to the duplet frequency. For the rest of harmonics, so the other frequencies related to the other subdivisions, there was a decrease. This could be maybe linked to some kind of a stabilization of the beat and its first harmonic. To compare across frequencies, we did a subtraction in which we took the pre-subdivision amplitudes from the post-subdivision amplitudes. And we ran a three-way mixed design Anova, and what we found is that there were increases at the frequency of the beat and its first harmonic, and decreases at the other frequencies. And the increases at the beat at the first harmonic were greater than the differences found for the other harmonics, for the triplets, quadruplets, and quintuplets. So no matter the subdivision rhythm, there is increases at the frequencies of the beat and its first harmonic, which corresponds to the duplet subdivision. Now, let's turn to the second comparison. Here we ran 5 three-way mixed design Anovas for each frequency of interest, and we found many main effects, interactions that were significant. And here I will just focus on the interactions that involve musical training and task, and sometimes also subdivision rhythms. So to start with, when participants were listening to the subdivisions, the duplets, triplets, quadruplets, quintuplets, there were no differences between musicians in white and non-musicians in gray. As you can see, in the listening task, there are no differences. However, when participants had to tap, differences appear. You can see that musicians in white had higher neural amplitudes compared to non-musicians. And this is across frequencies and also across subdivision rhythms. Interestingly, notice that at the frequency of the beat, 125 Hertz, there are no differences for musicians and non-musicians for quintuplets, to tapping to the quintuplets and tapping to the duplets. Maybe this is because non-musicians benefited from the duplets when tapping. Another way to do the comparison is to split the both groups, non-musicians and musicians, and see the differences between listening to the subdivision and tapping to the subdivision. And what we found is that in musicians, tapping to the subdivision made increase the neural entrainment to all subdivision rhythms and across frequencies, across the 5 frequencies of interest. However, in non-musicians, we can see that the tapping only increased the neural entrainment for duplets and triplets, while for the quadruplets and quintuplets was not significantly higher than listening to the subdivisions. Again, as before, we did a subtraction. Now we removed the listening to the subdivision from the listening and tapping to the beat of the subdivision. We ran a three-way mixed design Anova, and we found an interaction of frequency, rhythm, and group. I will try to summarize it here. First, looking at non-musicians, we found that the increases for duplets were greater than the increases for quadruplets, but there were no significant difference between increases in neural entrainment. So there in this tapping minus listening to the subdivision subtraction. However, in musicians, we found that there were increases And these increases were much greater for duplets, triplets, and quadruplets than quintuplets across different frequencies. So to summarize a bit our results, found that musicians performed better tapping, except for quintuplets, and also that non-musicians tapped more consistently to the beat of duplets compared with other subdivisions. We also found that after listening to whichever binary or non-binary subdivision, neural entrainment to the frequency of the beat and its first harmonic increased. And finally, we found that musicians and non-musicians' neural entrainment didn't differ when they were listening to the subdivisions. It differed when they were performing the finger tapping tasks that were predicting the beat. So from these results, we can discuss a little bit. And first of all, is there any subdivision advantage? And well, as you saw, non-musicians tapped better to the beat of duplets than the other subdivisions. So the neural entrainment to the beat was no different between musicians and non-musicians when they were tapping to the beat of the duplets. And finally, we found that also that first harmonic increased after listening to all the subdivisions. So after listening to all subdivisions, you go back to beat perception, but you increase the first harmonic. So all this seems to point that maybe there is a binary advantage or binary natural preference for binary structures in time perception, in rhythmic perception. This could be related to the well-known TikTok effect, or maybe also the evolution of bipedalism as humans. But also the 1 to 2, this binary subdivision is a universal found across musical cultures, as Sabai showed in 2015. And also it appeared in several experimental task reporting, including repetition of rhythms like Ravignani and colleagues did, in which some binary subdivisions appeared emerged from completely disordered rhythms. Also, importantly, there is a new study that Cecilia, Jan, and I ran lately, and we also found that the smaller unit to structure beat perception is grouping subdivisions in groups of 2, so in binary subdivisions. And I hope that it will be soon published. But well, some caution about the third statement, because regarding the first harmonic increase, the study by Peto, David, and James found that there was also an increase of the first harmonic when the preparation of the tapping occurs. So they also found that this same effect, but they linked that to the attentional demands of their task. So we don't know if this increase is just for stabilize the beat, so just pushing also its binary subdivision, or maybe it's some motor preparation attentional demands that interact with that. Of course, as all studies, we have some limitations that can lead to future research. The first one is that we don't have a beat tapping task, a one sound, one tap comparison to see that, okay, there is a subdivision benefit here. But the Zendel and colleagues in 2011 used a very, very similar inter-onset interval of 700 milliseconds, and they found a subdivision benefit. So we could say we're kind of safe on that sense. There is a very intriguing fact. So when participants were listening to the subdivisions, we couldn't find a clear peak at the frequency of the beat, except for quadruplets. This could mean that maybe the beat was not there, and maybe the subdivision was perceived as a new beat. So they jumped from a slow beat to a very fast beat. If this is true, then this has some implication. This may mean that we need some kind of external cues to anchor where the beat is, and allowing us to put some order and organizing the grouping of the subdivisions. Maybe external cues like as movement in dance, or amplitude modulations or pitch alternations as music. And finally, there is the issue of tempo. Quintuplets were faster than duplets. This means we kept constant the intertapping interval, always at 800 milliseconds. So if we added more subdivisions in that, they became faster. And unfortunately, the quintuplets inter-onset interval was 160 milliseconds, which may have crossed the benefit cost transition point that Reb talks in his studies. So we have to also look at that. Take-home message, musicians seem to have higher neural entrainment than non-musicians when they tap to the subdivided beat, when they are doing this predictive sensorimotor task. And also we found that there are neural responses that reflect an advantage for binary subdivisions, as found in the tapping responses. So if you are interested in more details, please look at our preprint in bioRxiv and send me a message for any question you have. And thank you all for listening. Thank you all my colleagues from the Music in the Brain and Center for Brain and Cognition, especially Boris, Juan, and Peter. And also thanks for all the funding and the collaboration agreements that allowed this research to happen. So now is your time if you have questions.
Thank you, Sander. Yeah. So now if anybody has question, they can just unmute and ask the question. Yeah.
I guess I have a quick question. This is a wonderful, oh here, let me do video. Really wonderful project, and I was really interested to see the results. I was especially struck by the dramatic difference between the musicians' entrainment when they were tapping and when they weren't. And just to be clear, you'd see much more entrainment to the subdivision when they were tapping the beat. Is that right?
Exactly.
Yeah, so do you
the
see that?
Subdivision and to the beat, yeah.
Right. So do you see that as just maybe going into a different attending mode? In some sense, they're more attuned to what's happening in the subdivisions when they're being called upon to do some kind of action based on it? How would you interpret that difference between both the conditions and the musicians, non-musicians?
So what seems to happen here is that it's only when listening, so when perception is involved, there are no differences between the neural entrainment that they put. And of course, it's more demanding to do a tapping task, a sensorimotor synchronization task. So there is some kind of attentional demands here, and they may influence. Maybe musicians are more engaged than non-musicians. That could also happen. But the split here is like, look, when non-musicians tap to the beat, there is only increases for the duplets and the triplets at the beat frequency and nothing else. But for musicians, there are increases at all frequencies for all subdivision rhythms, which means that they may have learned somehow to, sorry, oops. I wanted to share it. Let me show it like this. They may have learned to expand so musical training may have helped them to expand the advantage of using information, temporal information. Like, because you have more subdivisions, so you have more temporal information to use to be able to predict when the beat is happening.
They're only bringing that into play when they're actually being called upon to act in the context of these rhythms.
So how the task went, it was like they listen the beat, then the subdivision, the beat, and there is a countdown. So there is on the screen they see 3, 2, 1, and now they have to stop tapping the beat. So they have to keep the beat that we were listening before using the subdivisions. So this attention, of course, it could have interacted with musicianship as well, yeah. But if you want, please ask me more, because maybe I'm not answering you.
I'll let other people take questions. There's a lot to think about.
Okay.
Yeah. So we have one question in the chat by Jessica. So does tapping consistency correlate with Eeg peak amplitude at any of the frequencies during either listening or tapping conditions across individuals?
So tapping consistency correlated with the
Eeg peak amplitude.
I think I have here after the statistics. This is with the MET. Here is the peak amplitudes and the tapping consistency. Okay, so these are some correlations. And yes, for duplets, triplets, and quadruplets, it did at the tapping frequency, so the beat frequency, but not for quintuplets. There were no correlations between the amplitude increases and the tapping consistency. For quintuplets, it was not correlated, but for the other subdivision rhythms, yes, as I can see, and also across other frequencies as well. I'm sorry if I'm not sure if you can see the plot.
You can zoom it. Maybe you can share as a slide show.
Yeah, you can find this in supplementary material in the bioRxiv. These plots are there. How can I zoom that?
Okay. Yes. So we have we can take one more question. So we have another question by Jonathan. Yes. So I'd be very curious to see measures of tap to tap error correction and whether those are correlated with any EEG entrainment measures.
Again, please, sorry, I couldn't hear.
Just
Uh,
that I would be really interested to see the kind, for instance, autocorrelation or other measures of how much the error is corrected from one tap to the next. Which is known to be stronger with, I think this is one aspect of the subdivision advantage, is stronger error correction. And so I don't know. I think this could be a really rich data set for looking at that in particular, which I'm very interested in.
So
Just a comment.
to compare, I'm interested. So to analyze from one tap to another, if there is an improvement.
Yeah, so one thing you can do is look at the autocorrelation of consecutive asynchronies, and then another thing you can do is fit a model that assumes linear error correction and see what amount of linear error correction fits the tapping pattern best. And I think that it's connected with the overall precision of tapping, but it's not identical, and it could be interesting to look at that.
Yeah, I agree completely. Yeah, I haven't thought of doing that, but it's a good point. Yeah.
Are there any questions, or we can move to the next talk? Thank you, Xander. It was a very good talk.
Yeah. Okay, thank you so much. It was a pleasure. And stop. Mute myself.
So our next speaker for today is Tanya Gupta. So let me introduce Tanya. Tanya is interested in understanding the neural mechanisms underlying interval timing. In her bachelor's honors thesis, she investigated the effect of skewed probe distribution on temporal bisection in rats. She is currently pursuing her PhD in behavioral neuroscience from the Basic Behavioral Processes Lab, Arizona State University, where she is working with Dr. Federico Sanabria. So please join me to welcome Tanya for today's talk. You can start sharing. Yeah.
Sounds good. Okay. That is working. And let me just move. Okay, thank you so much for the introduction, and thank you for having me. I'm excited to talk to a bunch of timing people today about timing. So, as Mukesh said, I'm a PhD candidate in the Basic Behavioral Processes Lab at ASU, and I study interval timing. So a little bit of a brain shift from what Sander just talked about, so bear with me here. So just to give a little bit of an outline of what I'm going to talk about, give a little, I just pressed the end button on my keyboard, which is right by the, there you go. Okay, so I'll give a little bit of background on interval timing and the type of work that we do, and then think a little bit about the role of the hippocampus in interval timing, which is kind of what got me interested in studying longer intervals and studying the structure of longer intervals. And then think a little bit about the role of interval length in what is going on with interval timing here, and then move into a procedure that I'm implementing to tag components of behavior entrained to long intervals. So that's what we'll talk about today. So just to orient everyone, we're all here for timing, but of course that's broad, and Sandra just talked a little bit about more short millisecond timing. Our lab and I study interval timing, which is timing in the seconds to minutes range. So interval timing is from about the 1 second range to several minutes. I suppose the definition is flexible, but that's how I think about it, from about 1 second to several minutes. And this is distinct from those very short kind of millisecond or second level intervals that, for example, Sandra was talking about, or rhythm people talk about, and then also distinct from the very long ones, in circadian timing. And that's where interval timing lies, and that's what I'm interested in. And I think because of this like middle range that it's in, it's very psychologically interesting. So interval timing is actually implicated as more of a subcomponent of many cognitive processes, including decision making and foraging. One example that I really like to give to people is a hummingbird. So hummingbirds eat food by going to visiting flowers and taking nectar from flowers. And it turns out that they actually visit flowers at these like relevant intervals. So they understand the interval with which the flower's nectar is going to replenish, and they visit the flowers at these intervals that are beneficial for them, that are going to ensure that they get the most food possible. It turns out they can actually time multiple of these intervals at once. So that's a great example of interval timing in action. And this is implicated in a lot of animal foraging, but also just in conversation. So like right now, I know how long I've been talking. I know that if this was a conversation, that this would be an absolutely inappropriate amount of time to be talking without somebody else responding, right? So we have this sense of, how much time has passed, and that's what interval timing is. And in the lab, and in our lab in particular, and in many interval timing labs, we use rodents, and we train rodents to seek food at specific times by pressing a lever. So that's how we study this phenomenon in the lab. Another interesting thing about interval timing is that I would argue that the neurobiology of interval timing is something that we're still figuring out. So a precise like circuit for interval timing hasn't been described, and certainly some people say that there might not even be one, but I won't get into that. But one brain area of interest that I want to focus on is the hippocampus. You're probably familiar in some way with the hippocampus. The hippocampus is, of course, implicated in the encoding of long-term memories. And what we know about the hippocampus when it comes to timing is that it is involved in timing. That's about as much as I'm comfortable saying with like some level of certainty, is that it is involved. Again, maybe some people would have more to say about that. We know this at the neural level because there are identified time cells in the hippocampus that track time between events, such that when you sort them by peak firing rate, like is done here, they have a unique peak firing time. We can also see this at the behavioral level, which is what we're going to focus a little bit more on in this talk, in lesion studies. So for example, a common way that we assess interval timing in the lab is by training rats to seek food after a specific fixed interval. So if you press the lever after 10 seconds has elapsed, then you will receive food. And this graph shows the normalized rate of responding as a function of time within a trial. And animals that have hippocampal lesions, this is post-lesion right here, so we lesion the hippocampus, we actually injure the hippocampus, they have earlier peak responding rates. And this earlier peak responding rate is a pretty reliable and replicable result. And it's one that we investigated in our lab, which is what I'm going to talk a little bit about now. So the way we did this in our lab, in the study I'm going to talk about now, is using a task called the switch timing task. So in the switch timing task, animals are trained to seek food at 2 different levers, each of which delivers food on a different program schedule. So one of them is programmed on a short fixed interval, or short FI, which means that the first lever press that occurs after a given shorter interval has elapsed is reinforced. And the other lever is programmed on a long FI, or long fixed interval, which delivers food at the first lever press that occurs after a given longer interval has elapsed. I'll get into specific intervals in a minute, but the important thing is that even though in each trial the animal has access to both levers, only one of these schedules is active in each trial, which means that only one of these schedules will deliver reinforcement when a schedule is completed. So even though the animal has both levers, only one is working, but we don't tell the animal which of those levers is working. So as a result of this arrangement where only one of those schedules is active, but we don't tell the animal which of the schedules is active, what we expect on trials where the long FI is active is that animals will start by seeking food on the short FI lever, because that potentially delivers food sooner. And then after enough time has passed without receiving reinforcement, they will switch to seeking food on the long FI lever. So this gives us a few indices of timing, but the one I'm going to focus on here, just for the sake of brevity, is the latency to switch, or LTS, which is the time of the first lever press on the long FI lever. So when does the animal start seeking food on the long FI lever? And in this case, we're going to look at the median of that measure as a means of assessing how the hippocampus is involved in that process. So in our lab, we did a study where we trained a bunch of rats on this task, and we trained animals with a short FI of 8 seconds and a long FI of 16 seconds. And the animals were split into two groups. The animals either received a sham lesion, so just a fake surgery that was that did not impose any lesion in the hippocampus, or received excitotoxic NMDA lesions in the dorsal hippocampus. And if we take a look at the LTSs, when we did this, the rats with NMDA lesions initially switched to the long FI levers significantly sooner than did their sham counterparts, but that this recovered as training continued. So as the animals were trained to reacquire this 8-second, 16-second switch timing task, the lesioned animals eventually caught up or ended up at similar levels to their control counterparts. So the LTSs were shortened as a result of the dorsal hippocampus lesion initially, and this mirrors kind of what we often see, what I talked about earlier in hippocampal lesion timing studies, where the lesion shifts the time of responding to a time earlier than it was prior. So following that phase, we move the rats to a new phase in which we increase the length of the long FI to 32 seconds. So now the short FI was still 8 seconds, but the long FI was now 32 seconds, which is quite a bit longer. And we looked at the LTS in this condition. And in this condition, dorsal hippocampus lesions resulted in significantly shorter LTSs, and this was sustained even in asymptotic performance. So even as we continued to train the animals on this condition, the dorsal hippocampus lesion animals continued to have these shorter LTSs, switching to that long FI lever sooner. So what we observed in this study is that the shortening of LTSs was enhanced and sustained when the long FI was increased to 32 seconds. And that observation was very interesting to me. There were many results in this study, but kind of this observation is what is driving a lot of my current research. This observation that encoding a new interval, and particularly in this case, a longer interval, which is something that is interesting to me, is what's driving some of the work I'm doing right now on my dissertation. And the reason for that interest is not only because like the length of the interval itself, which of course is interesting because we're interested in how intervals are timed, but when I started to think about this more, I started to think about all of the features of the testing environment that change when we increase the length of the timed interval, because of course timing doesn't happen in a vacuum. So we have to think about all of these things that change when we increase the length of the time interval. So now we're asking the question, how can we test the role of the hippocampus in encoding these increasingly long intervals? Because when we think about it, for example, when we increase the interval length, and when we train animals in interval timing, we're essentially teaching animals about a delay, right? We're teaching animals about a delay to reward. And when we increase the interval, we're increasing the delay to reward. And we know from the very, very dense and rich kind of delay discounting literature that delay devalues reward. Delay can be aversive, perhaps. And so when we increase the length of the interval, we have to take that into account as we've devalued the reward, maybe. Another thing, if let's say we've potentially devalued the reward by increasing the length of the interval required to get it, one thing that we've maybe done is induce a decrease in motivation. So we know this because when we ask animals to initiate their own trials, which we do very often in our lab, the time, and look at the time it takes them to initiate those trials, the time it takes the animals to initiate those trials is increased when the to be timed interval is increased. And this could reflect a number of things. such as different aspects of behavioral systems, but it could also reflect a change in the motivational state of the animal, suggesting that a change in the global rate of reinforcement as a result of increasing the interval length reflects in a change in the animal's motivation to actually engage in the task, separate from its timing ability or timing capacity. And then the last thing I want to mention is that when we increase the interval length, we also undergo an increase in non-timing behavior. So in our lab, we do some computational modeling to try to see how behavior is structured on a response by response basis. And when we measure behavior in timing tasks, we know that the totality of behavior is composed of both responses that are adherent to timed responses, and behaviors that aren't adherent to time, right? Sometimes the animals have other things to do, and I understand that. So if we model that, we know that the proportion of non-timed behaviors increases as we increase the length of the interval. This could, again, reflect motivational, attentional, reward, incentive, salience processes. So if we think about all this, when we increase the length of the interval, we've changed all these things about the testing environment, potentially increasing the delay to reward, potentially decreasing motivation, and potentially increasing the proportion of non-time behavior. It's clear that there's like a number of experimental concerns to address if we want to study these really long intervals, which I am interested in doing. So in order to kind of set this stage to study these longer intervals, there's two task adjustments that we propose, that we want to implement to study interval timing tasks in a way that takes these different aspects into account, especially when we're studying these increasingly long intervals. So the first is that subjects should be afforded with the opportunity to engage in reinforced non-timing behavior. So present the animal with a reinforced non-timing alternative so that we can tag that non-timed behavior and understand how it is structured concurrent with timing. And then we propose that subjects should initiate the onset of time stimuli on their own. So animals will have to initiate each trial on their own, and this way, any interference with motivation would be detected in the rate at which these time stimuli are initiated. So if less trials are initiated or the time between trials initiated changes, we can interpret this as a change in the motivational state of the animal. So we're able to assess multiple things at once. So since we've established that timing data generated from animal models has this inherent motivational dimension, this delay to reward, one way that we can get around this, or at least study it, is by using these response-initiated schedules of reinforcement, where the initiating response that starts the trial is separate from the actual reinforced response that, in this case, is a timed response, right? And these work pretty well. You know, we implement them in the lab a lot. But one thing that we know is that when we try to test these longer and longer intervals, and we ask the animal to initiate every trial on their own to start, the task, we see an increase in schedule strain, what we call schedule strain, where the animals decrease the number of initiated trials, especially at these longer delays, and this yields less analyzable data for us, and potentially the data that are available are polluted by this change in motivation, right? And so one way that we thought that we could get around this, or at least facilitate responding even at these really long intervals, is the timing with opportunity cost tasks that was implemented previously in pigeons. And we implemented a response initiated version of this task in rats. So that's what we're going to talk about now, is how I went about implementing this task in rats and having the rats initiate the trials themselves to study this structure of long time behavior. So in this task, animals had to initiate each trial on their own using a nose poke response. That's what NP stands for. So the animals had to nose poke a nose poke device, and this initiating response serves as our index of motivation, where the frequency of these initiations serves as an index for the extent to which the animal is motivated to engage in the task. When they do this, the 2 levers come into the chamber, and each of those levers is programmed with a different schedule. The first of these schedules is a random ratio or RR schedule, which is a probabilistic schedule, which means that food is delivered with a low constant probability. So it's not based on time, it's based on probability. So for example, in a random ratio 5, every lever press has approximately a 1 in 5 chance of being reinforced. So in this case, the subjective probability of reinforcement across responses is constant with training. The other lever that the animal has access to is a fixed interval lever, which is the same as what we saw in the last task, where the first press after a given fixed interval has elapsed is reinforced. And in this case, we know that the subjective probability of reinforcement as a function of time increases. So in this way, in these trials, both levers are available, but again, we like tricking animals in our lab, and only one schedule is active and the rat is not cued as to which. So because of this arrangement, what we expect in terms of presses as a function of time is that animals will spend most of their time hanging out on the random ratio lever, and then will increasingly check that fixed interval lever as the fixed interval approaches. And so that's what we expect about the structure of the behavior. And so in this way, we're looking at timed responses, right? Because we're looking at the responses on the fixed interval lever. We're looking at tagged non-time behavior because we have behavior on the random ratio, and we can observe how this changes as a function of any of our treatments compared to how the time behavior changes. And then also we have an index of motivation because animals have to initiate each one of these trials on their own. So this is a pretty elaborate setup. And so I spent some time this past year trying to see if we could get rats to do this, and to do this at increasingly long intervals. And so we tested this on 6 rats, and I'm going to show you kind of what that looks like. So we implemented this task in a few rats, and what we found is that we did observe a pattern of responding similar to what we expected given the structure of the task. So here we're looking at raster plots of the response structure from an individual animal where the random ratio schedule was active. So here the random ratio schedule was the one that delivered food, but the animal had access to both, right? So they were still going to be checking that fixed interval lever. So, and here in red are the responses on the fixed interval schedule, and in blue are responses on the random ratio schedule. So here in an FI 12 seconds condition, we can see that the responses on the FI lever are mostly clustered around this 12-second mark at the dashed line here. But we also tested this using two longer schedules, the 24-second and 48-second schedules, and we observed a similar pattern here. And so the important thing to note is that the animal initiated each one of these trials on their own. So it seems like they continued to initiate these trials even at an increasingly long delay to reward, which is our goal here, is to be able to tag behavior in this way and get animals to initiate trials here. We can also look at peak functions, which are a very common way of visualizing responses in timing tasks. So on the x-axis here is the proportion of the fixed interval, where 1 is the fixed interval itself, so in this case 12 seconds, and 2 is twice the fixed interval, so in this case 24 seconds. So we can look at all the responding, and on the y-axis is the response rate. And it looks like the rats are responding maximally at the reinforced time. And this also appears to be the case in longer FI conditions if I equate bin sizes. So by using this task, we tagged non-timing behavior, we gave the animal the opportunity to engage in that reinforced non-timing behavior, and animals continued to initiate trial at the 48-second condition. And importantly, behavior on the FI schedule still appears to be under temporal control, which is what we were concerned with. We also finally ran a probe where we removed the fixed interval, sorry, we removed the random ratio trials types. So we removed the opportunity to engage in that non-timed reinforced behavior. And we did this, and even though the data are noisy because we had 6 kind of very old and somewhat haggard rats at this point, it seems like the frequency of trial initiation did decrease. It's a little bit noisy when the opportunity to be reinforced by the random ratio schedule was removed. So the presence of the RR schedule facilitated trial initiation to some extent, and we're doing this now with many, many more animals. So hopefully we'll understand that a little bit better in the future. So that's the data I have right now, but having established this procedure a little bit, I am working on my dissertation, implementing this procedure and pharmacologically deactivating the hippocampus so that will allow us to elucidate whether any changes that we see as a function of interval length is related specifically to timing or to motivation or the structure of non-time behavior, so we can get at that in a little more of a detailed way. And with that, this is my lab. This is a very outdated photo of the lab from pre-pandemic times, and our funding sources, and Twitter if you like Twitter. And I'll take questions.
Yeah, thank you, Tanya. That's a very good presentation. And if anybody has a question, please go ahead and unmute and ask, or type in the chat box.
I can ask a question. So Tanya, that was a really great talk. So my question is, I know that you specifically talked about the latency switch on long trials only, but if I'm not mistaken, you haven't talked about the variability of those switch times or response rates overall under NMDA lesions in the dorsal hippocampus. Could you maybe talk a little bit about them?
I definitely can. So,
and also, sorry, and how if they differ as a function of training, basically.
As a function of training? So
I mean, it's training length as a function of session number. Because you could see an increase in accuracy, basically, but what happens to the trial variability, for instance?
Yeah, so on the bottom here is the coefficient of quartile variation of the LTSs, and this is following the lesions. And so you can see initially, maybe one trial, the lesion animals were a little bit more variable, but this didn't persist very long. And in baseline, I can find the graph if you want, but in baseline there was no difference. So we throughout this paper, which is published if you're interested, that was a theme that we found where animals' central tendency measures were affected by the lesion, but not the variability measures in this case.
And well, that's really interesting. And so the black one is, wait, I'm also interested in the data to depart. So you're not really, so that's on the top. Okay, gotcha.
That's the variability and latency to depart. And actually the central tendency of latency to depart was intact.
Yeah.
As well. Like in this study, we saw like specifically an effect on the latency to switch, and that was very consistent across conditions.
All right. Okay. Thank you so much.
Yeah, no problem. Thank you.
Is there any more questions, or I can ask one question?
Yeah.
So, like, so you mentioned that you have the sham lesion conditions. So how do you do that? Like when you say sham, do you like open the brain region and then just don't make a lesion, or like what do you do?
So we go through the whole thing except for infusing the drug that does the lesion. So in this case, the lesions were done with essentially an acid that does the lesion. So we go through the whole thing. We lower a needle into the hippocampus and everything. We just don't infuse anything. So they have everything, and they even have sometimes like a small needle track or something, but they don't get an actual lesion to do everything.
That's a very nice control. Like that's a good one. And second question I had was like, you also mentioned that tagging the non-timing behavior. So can you talk more about that? How do you do that? Like, so I didn't understand that part, because for me, the behavioral is everything is timed. So how do you separate out that?
So in this case, the non-timing behavior was the random ratio schedule. So here, the random ratio schedule is based on probability and not time. So the animals are reinforced based on probability, where every lever press has a 1 in X chance of being reinforced. So it's not timing based. So across time, on the random ratio schedule, the subjective probability of reinforcement should be more or less constant. So that's not a time schedule. So by looking at that compared to the fixed interval schedule, which is timed, that's how we can compare timing and non-timing behavior in this task. You could define non-timing behavior, obviously very loose, like any number of ways.
Okay.
But in this,
got it.
that's what we're doing.
Thank you.
Mm-hmm.
Do we have any more questions? You can take 1 or 2 questions. Okay. I think that's it. I think you explained very clearly. It's a very good presentation. So now I think Lucilia will share the slide for the next, like the journal club. Yeah. So in the next journal club, we will have Sandeep Teki, and the more detail will be announced later on in the emails. But that's what we have for now. And we also have put links in the chat for, like, if you want to nominate anybody for the next speaker, you can like fill the form, and you can also subscribe to the newsletter so that you get regular updates each month. Okay. And I would like to thank everyone for being present for this talk, and thank you very much to the speakers. Thank you, everyone. Yeah, I think we can conclude for today. Have a good day, have a safe day, and get vaccinated.
Thank you.
Thank you.
Thank you so much.